My name is Daniel Gomez. I'm going over precision immuno-oncology in the Snyder Lab at Department of Genetics, Stanford Cancer Institute, Stanford University School of Medicine, and Department of Biological Sciences, Biology in CSU East Bay. Excuse me. All right, so first I wanted to start with immune checkpoint inhibitor-based single-cell RNA-seq data sets. There's an extensive literature review and meticulous performed data curation, quality control, pre-processing, and analysis, and a robust data resource comprising of 8 single-cell RNA-seq datasets spanning 9 distinct cancer types involving 223 patients, 90,270 cancer cells, and 671 other cell types. In this ICI-based single-cell RNA-seq datasets, we see a single-cell data housed that's user-friendly, and the cell-to-cell interactions between malignant cells and other cell types with incorporated other cell types. So the cancer types are basal cell carcinoma in Yost et al., hepatocellular carcinoma in Ma et al., intrahepatic cholangiocarcinoma in Ma et al., and melanoma in Arnon et al. Next is, or the fifth one is, so that was one, two, three, and four. The fifth one is melanoma brain metastases in Breckenridge et al. And HER2 positive in Bassez et al. And then you have triple negative breast cancer in Bassez et al. The eighth one is positive Bassez et al. And clear cell renal carcinoma in B or by et al. So this is in Gondol et al. 2025, where we have 9 different cancer types in 8 different studies, 223 samples, and 90,270 malignant cells, and 265, 671 other cells. So what I did is I went into the shining cell that was built, and we have single cell pre-post treatment cancer type, RNA contribution, and CD74B2M interaction map. So here's the interaction map that I was able to show, where we can see malignant cells, fibroblast, HPC-like plasma cells, TAM or TAM cells, TEC or TEC cells, T cells, B cells, mast cells, um, CAF cells or cancer-associated fibroblasts, as well as next to fibroblasts, and plasmacytoid dendritic cells and myofibroblast. So those are all the different cell types that are shown in the UMAP, uniform manifold approximate projection. And next to it, we see the different cell types. So in a UMAP, we can see all the different 9 different cancer types. So we can see HER2 positive, T and B, C, ER positive. So 3 different breast cancers, sub cancer types, and MBM, melanoma, different skin cancers, metastatic melanoma, uh, kidney cancer or renal cell carcinoma, and then HCC, which is hepatocellular carcinoma, and ICCA, which is the other liver cancers cancer type. Over here, we can see the expression of specific transcripts in the cell. So we have single cell RNA-seq and the transcript RNase-1 shown here for melanoma. And then for HCC or liver cancer, we see CCND1 and GPC3. So that's what's shown there. And then CD74 and B2M interaction, this MHC complex is interacting with different TCRs in the immune system. We can see the expression of that interaction, high and low, shown here and here in a UMAP, as well as another heat map. So we can see RNase-1, GPC3, and CCND up and down regulated. I went ahead and took the single cell RNA-seq dataset into Liana for cell-cell communication and ligand receptor interaction machine learning framework analysis. And we can see the different sex of the donors. So male is over here in orange, female's in blue. And then we separated out the disease or cancer cell type. We can see cancer cells by their cell type, and I would look for here hepatocellular carcinoma in pink and blue, intrahepatic cholangiocarcinoma. So we have two different cancer cell types in here near the middle. Pre and post single cell ICB immune checkpoint blockade. We can see before and after treatment. So orange is before, pre, and blue is post or after. And then next we see favorable and unfavorable responses. So we have favorable here in blue and then unfavorable here in green. And then in their outcome, you can see complete response, partial response, ICB partial response, ICB stable disease response. So here we see, you'd be able to analyze partial response here in blue. Um, let's see other letters here. Majority I'll have to look up. There's E and R as well as NE. So E and R and NE. Usually we do complete response, partial response, stable disease, or non-response. Here we see non-response in brown. Here we have next we have the treatment. So the treatment is in purple. We can see a neoadjuvant and pembrolizumab together here, as well as pembrolizumab alone. So neoadjuvant and pembro, and then pembro alone here in the top right. And then let's see, in brown we see ipilimumab and nivolumab together, and green we have ipilimumab by itself. Those aren't too common. I would focus on the pembrolizumab, much more effective. There's the temozolomide, pembrolizumab, and an angiopoietin 2 inhibitor with ipilimumab in pink. I see just a little signal over there. And then nivolumab. Nivolumab's well known. Here we can see it in blue. And then durvalumab and tremelimumab up here. And then prior to treatment in brown, nivolumab prior to treatment is a good one. So prior to treatment, seeing what it looked like before treatment, nivolumab, ipilimumab, ipilimumab, mumab, ipilimumab, and nivolumab together prior treatment, and then with pembrolizumab, neoadjuvant, and pembrolizumab together here in purple and brown. And what we want to do is all the cell types. So seeing which cell types they are, we can see here in this UMAP with Liana, T cells. We have an abundance of T cells, and malignant cells are over here on the other side. And in red, we see endothelial cells and fibroblasts, and lymphocytes over here in lime green, emerald lime green. And then below it is a heterogeneous cluster here where you see a lot of overlap, something that could be um, like it could need further analysis for separation. So clearly separated are T cells, malignant cells, endothelials, and um, and that's pretty much it for those. Next is if we can see it's a primary cancer or metastatic cancer. So primary cancer is here in green, metastatics in blue. The assay used, SmartSeq 10x 3 prime version 2, version 3, or 10x 5 prime transcriptional profiling. So here 5 prime transcriptional profiling for the most of it, and 10x 3 prime version 2 and SmartSeq here, and then some 3 prime version 3 over there. And then after we reanalyzed and went a deeper level into the cell types, and you can see um, Over here we have CD8 exhausted or X T cells. And we can get more into those cells and T cell exhaustion and cancer immunotherapy, as well as immuno-oncology and the cancer and what's happening in different cancers. Up here we have fibroblasts, endothelials, and CD8 memory T cells. So this is a good thing. We are able to see CD8 memory T cells that are able to mount an immune response once they recognize the cancer. And then up here we have endothelials, still at the same population number. And then here we have the fibroblasts, which provide structural support and are able to be remodeled after therapy. Over here is the myeloid cells, kind of like the innate immune system and different cell types. B cells are over here, and then some NKT cells, and like a newer cell type, NKT cells, both NK cell and T cell, a little bit of both arms of the immune system. So that was cancer immunotherapy, and here we have personalized cancer vaccines therapies, and the development of this is complex. What the complexity at scale is that the immune system has to fight the tumor and underlie tumor immunity to have successful induced anti-tumor T cell response in the human body. So there's a clinical trial landscape for personalized therapeutic cancer vaccines and the complexity of dosing strategy and dose response for personalized neoantigen-based TCVs. And immune cells and the PBMCs are acquired from patients detect an informed antigen-specific T lymphocyte response. So the development of these PCVs and TCVs we have to know the mechanism of action. We have to know the delivery system, the immune monitoring, the route, the dosing strategy, the algorithm for antigen selection, indication, adjuvants, manufacturing, combination, therapies, and obstacles and regulation. So these are all the different steps of development and complexity at scale. And this is going into future research. I'll go ahead and skip over this slide. Most of it is how it works, kind of describing the platform. So the immune system uh, underlying the tumor immunity and successful induced anti-tumor T cell responses in the human body. It's located in the lymph node and tumor. So what happens is you have a priming of activation in priming of activation where we see IL-2, CTLA-4, and PD-L1 and PD-1 interacting in the lymph node with these therapeutic cancer vaccines. So there's cancer antigen presentation. Cancer presents an antigen and it gets primed and activated and then trafficking of the T cells to the tumor. So the tumor goes through the blood vessels with the T cells. T cells go through the blood vessels and it's infiltrated in T cells into the tumors. So we have an infiltration step where T cells are diabetes from the blood vessels to the tumor. And the recognition of cancer cells by T cells in the tumor are there. So you have tumor cells and the recognition of cancer cells by T cells. And the killing of cancer cells happens with PD-L1 and PD-1 interaction. You can see that in there where you see the T cell interacting with the tumor. The tumor has the T cell has the PD1 and the tumor has the PD-L1. Release of cancer cell antigens um, could occur with chemotherapy, and that's when the cancer cell releases antigens, and then they're able to be presented like shown here on this DC cell. Other cell surface proteins are FASL, MHC class 1, class 2, interferon gamma, PDL1, and PDL2. and the TCR. And then it's showing mRNA. Let's see, mRNA here. mRNA is in the therapeutic cancer vaccines. You can also use peptide or proteins and cell-based therapy and viral vectors. So APCs are the antigen presenting cells. So we have APCs, tumor cells, T cells, and tumor antigen. I just took you through the whole immune mechanism for tumor immunity and successfully inducing an anti-tumor T cell response in the human body. So these are the schematics of a common TCV immune system to induce human anti-tumor T cell responses. Next, we'll go over in this schematic where therapeutic cancer vaccines have tumor antigen. And when they get released, you have tumor cell apoptosis or necrosis, and the APC in the tumor recognizes and presents the tumor antigen on the surface. And the APC moves to the peripheral lymph node through the lymph tube. And in the lymph node, we have naive T cells and lymph nodes and effector T cells. And the effector T cells and memory T cells have their own trajectories, and they home to the different locations. So effector T cells release TNF and interferon gamma, and those go into the tumor. And memory T cells interact with or become central memory T cells or effector memory T cells, and the TNF and interferon gamma are the effector function of T cells and leave the lymph nodes, moving to tumors location, targeting tumor antigen and killing tumor T cells or killing tumor cells. And that's sort of the mechanism of immunity as well that happens from the lymph node to the tumor. I was able to show you from the lymph node that's distal, distant from the tumor. So any lymph node can interact with the tumor to have a successful induced anti-tumor cell immune response in the human body. And for personalized TCVs, neoantigen vaccines are individualized and tumor-specific therapies, typically targeting multi-tumor antigens in unique patients in each unique patient. And we may identify or confirm non-synonymous somatic mutations expressed in the tumor for inclusion of personalized TCV, a biopsy, and the tumor taken for a whole exome and RNA sequencing. Mutations are analyzed using MHC class 1 epitope prediction algorithms, and those are prioritized. So we have a variant mutations that can be targeted by neoantigen-based vaccines. So these include single nucleotide variants resulting from a single nucleotide change from one base to another indels as insertion or deletion of sequence of nucleotides from genome, resulting in frame shift mutation that may alter protein function. Ranked list and candidate antigens are further refined, and we can do in vitro binding assay results and with synthetic peptides that are tested and binding for appropriate class human leukocyte antigen allele of interest. And neoantigen selected may be of clonal origin and present to tumor cells or clonal, which are present only in a subset, both of which influence immunoreactivity. Mutations can also occur classified in their role of tumor growth and passenger mutations lacking intrinsic growth advantages, and driver mutations that provide growth advantages selecting during tumor evolution and incorporating into personalized TCVs. In the clinical trial landscape for personalized TCVs, we have 23 personalized TCVs currently in phase 1 or phase 2 from 13 major sponsors. Similar to Genentech and Roche and um, uh, other 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 major biotechnology companies. This analysis of personalized TCVs in clinical trials were performed to investigate the various platforms, antigens, algorithms, delivery systems, clinical settings, endpoints, and primary clinical findings. Sources include clinicaltrials.gov, company websites, press releases, conference presentations, and abstracts within the last 5 years of American Society of Clinical Oncology, ASCO, European Society for Medical Oncology, ESMO, and Society for Immunotherapy of Cancer, SITC, as well as the American Association for Cancer Research, AACR. In addition to PubMed search for relevant publications. So we're able to see the trials per sponsor and type of vaccine, if it's an mRNA, peptide, Listeria, bacterial, DNA, mRNA, viral vector, yeast. Among these trials, 20 of our phase one investigations with 3 trials per sponsor, with most using mRNA, DNA, peptide-based antigen delivery platforms. And the bulk of the trials are enrolling patients with multiple solid tumor types. Trials are in non-small cell lung cancer and melanoma are the next most common personalized TCV trial indications. And many of the TCVs are being tested in combinations with other treatments, including chemotherapy, interleukin-2, and anti-program death PD-1 and PDL1 uh, program death ligand 1, given the single antigens in combination with anti-cytotoxic T lymphocytes associated protein 4 or CTLA4. Immunogenic platforms must, 1, benefit broader patient populations, 2, are comprised of fewer antigens, 3, offer less frequent immunization schedule, 4, require a less invasive delivery route, and 5, most amenable to combinational approaches that hold practical advantages for later stage development. So we can see these pie graphs, the phase of the trial, the number of antigens, the indication, the combination partner, route of administration, delivery system, vaccine administration frequency, vaccine doses per year, number of vaccine dose levels tested. Now, we went ahead and retrieved some clinical trials that were applicable to our research. Um, that had CRC, colorectal cancer, lung cancer, non-small cell lung cancer, pancreatic cancer, MSS, CRC, EC, and BC. Um, so endometrial cancer and breast cancer, as well as lung cancer and colorectal different uh, uh, colorectal cancer. There's also bladder cancer, and the regimens for each are tumor-associated antigens, mRNA, that targets KRAS mutations, and neoantigen adenovirus vectors help amplifying mRNA edge, novel AI model for antigen prediction, and optimal immunogenicity, T cell recognition, intramuscular via viral vector, um, using a D Chad V prime 1 by 1012 VP SAM boost. That's 30 to 300 micrograms. So that's an adenovirus, viral delivery. And then we have neoantigen tumor-specific shared neoantigen peptides, peptides with heat shock proteins with HSP70, as well as 240 auto Synvax and 50 microgram QS adjuvant Q2W up to 1 for different cancer cell types. That's in phase 1, and that was terminated in 2019. And then phase 1 active not recruiting 2020 completion. We have Ad Advax immunotherapies and Amgen that use a neoantigen Listeria monocytogenes secreting TAAs in a mind system for with ML or machine learning to predict and weigh rankings in the allele frequency, tumor drivers, MHC binding, infusion. That's 15 minutes attenuated bacterial vector in lung cancer, colorectal cancer, head and neck cancer, um, and urinary cancer. And there's only 5 patients in that. So that's phase one. Another phase one with 28 patients, as they were able to use the same bacterial tumor-associated antigen, Listeria monocytogenes for and just focus on colorectal cancer. And then phase one recruiting and for NAT bioscience, neoantigen yeast-based injectable suspension recombinant yeast-based vector, CRC, BC, HNC, NSCLC, pancreatic and liver that had 16 patients. So these are early phase one trials um, that some recruiting and not recruiting. Other ones are complete. So we'll go over the dosing strategy next. So for personalizing antigen, the complexity of the dosing strategy and dose response, it's limited. So the limited absorption, distribute distribution, metabolism, and excretion or ADME is characterized due to a poorly conducted dose-finding trials that jeopardize the chance of approval of some personalized TCVs that might have otherwise provided clinical benefit to patients. So the first step is to use a new antigen encoding mRNA decatopes that are loaded onto nanocarriers, and we have patient-specific constant regions, and then patient-specific. We have mRNA in between lipids and a nanocarrier, and this goes into intravenous administration, rapid clearance to lymphatic organs. So we get the plasma concentration of the carrier in nanograms per milliliter and time post-infusion by hour. And that's for rapid clearance to the lymphatic organs. And then for the PK or pharmacokinetics, we can see the plasma concentration of the mRNA and the time post-infusion per minute. After that is the processing of neoantigens. So we can see MHC1 and MHC2, as well as the endosome and lysosome. So it starts with the mRNA. The mRNA is able to be processed by the ribosome and the proteasome, and then goes into the endosome and lysosome and presents MHC2 and MHC1. Next is the T cell stimulation. So you have tumor cell and T cell interacts, and the T cell is stimulated by the tumor, where we can see cytotoxic mediators, cytokines, intracellular cytokine cytometry. And then we are able to get the T cell response with variable functional avidity to antigens with the pharmacodynamics. So with an ELISA spot, we can count the ELISA spot T cell response as well from the peptide concentration. So the peptide concentration ELISA spot, we get the PD, and the T cell response with variable function avidity to antigens. The immune kinetics is shown in a dose antigen response relationship, where we have D1 and D2, D3 and D4 with percentage immune response. Lastly, we have the immune response relationship with the objective response and tumor regression. So we have the tumor type, dose level, and neoantigen count, patients, tumor response, and T cell response for where we can look at the immune response relationship with objective response to tumor regression. So that's the complexity of the components impacting dose strategy from dose response for personalized TCV. So the favorable absorption and distribution of antigen and coder antigen to lymphoid organs are precursors to a successful immune activation, anti-tumor CTL, and effective tumor killing. And we can see that immune monitoring techniques such as analysis of cell population by cell flow, including phenotypes, functionalities, and activation status, and enzyme-linked immunospot ELISA spot assays to measure cytokine release after antigen-specific immune responses, and peripheral cytokine profiling using an enzyme-linked immunosorbent assay, or an ELISA, to evaluate innate immune responses. Tetramer analysis using MHC multimers loaded with antigen peptides to measure antigen-specific CD8 T cells, and T cell receptor analysis by sequencing a polymerase chain reaction, or PCR, to elucidate the immune repertoire, including genetic arrangement and specificity. Requires careful evaluation of stability and assay conditions with use of proper controls to accurately evaluate induced cellular immune responses. So we have efforts that have to be taken to reduce potential sources and variable immune reliability as immune monitoring relationships in response to personalized TCVs routinely used to inform clinical decisions. More recently, the NCI, or National Cancer Institute, has supported an initiative to fund 4 cancer immune monitoring and analysis centers in support of research conducting state-of-the-art analysis such as immune profiling, characterization of responses in selective immunotherapy trials. So immune cells and PBMCs acquire from patients, detect and inform antigen-specific lymphocyte response, and they essentially function as surrogate biomarkers for efficacy. This is showing you the steps, step by step, what I went over. The tumor here. I'll go over the 6 steps for this efficacy surrogate biomarkers and immune cells and PBMCs. So we have tumor antigen identification, in vitro simulation with multimer incubation, peptide stimulation, peptide culture with T cells, analysis, analysis methodology, cytokine production, ELISA spot, fluorescent multimer flow cytometry, proliferation, and with TCR-seq, interpretation of results, routine measurement of treatment strategy for clinical care of a patient. And then that includes vaccine and combination therapy. Step 6 is immune-mediated anti-tumor response, where we can see the intracellular cytokine cytometry and the T cell that interacts with the tumor cell. And in the ELISA spot assay, we collect blood, isolate PBMCs, stimulate with antigen, incubate, deliver, and read spots. And this MHC tetramer assay, we have a MHC, streptavidin, and fluorochrome that are used in these and the MHC tetramer assay. So those are the steps of using patient PBMCs and detecting and informing antigen-specific T cell lymphocyte response, essentially function as a surrogate biomarker for efficacy. And I wanted to go over Immunomine, personalized cancer vaccine multimodal immune profiling repertoire analysis that I did. We can see the repertoire overlap in samples, different samples, over here in A. And then I did a Chow-1 sample density, where we can see the specific samples, which one had the most sample density estimation. And you can see that is over here. And next, lineage tracing or lineage tracking of terminal T cell exhaustion clones. So you can see the CDR3 epitopes here, and which ones have the highest CD8 T cells. And then over here we have a UMAP for each sample. Number of barcodes per sample and number of receptors per sample in each tissue and blood, normal and tumor. And the number of barcodes per sample in each tissue by the sample. Next, we have multimodal immune profile repertoire analysis, where we have TRB V21 and TRB V28, TRB V51, all in 3-1 MT tumor, or we can see the number of receptors and the proportion of them. So here we can get a clean readout of the gene usage for the multimodal immune profile repertoire. Same thing here. We can see the usage over here, but we see in for 4.6b Tregs and tumor, we can see the most TRBVs, and there's also some resident tissue, resident T cells in the tumor in 8.3b. And getting to the CDR3 immune repertoire length distribution by tissue, we can see a fraction of repertoires that are hyperexpanded in a large rare clone, where we can see them as hyper expanded, large, or medium for each tissue. And then the fraction of the repertoire top 10, 100, and 1,000 for the clonal rank bin. And then the CDR3 length distribution by tissue is shown over here, red for blood, yellow for normal, and green for tumor. And then the length distribution for the tissue resident memory and tissue. Um, so for different clusters, we have CDR3 length distribution for TRM and Tregs, and then the rank abundance for 1,000 clones per repertoire for all the different T cells and the clones. Here we have the coverage diversity, DXX, and we can see the IMD repertoire ID. So we see 3, 9, 23, 30, 34, 43, 50, all those different immune repertoire IDs for the coverage density and DXX. And then the Chow-1 richness estimator being used, we see mostly blood in the estimator. And then Shannon entropy bits, we can see all the different cell clusters. And in each cluster, we can see the amount of Shannon per cluster. And it looks like this TCF7 in blue has the highest, and then FOSS is right below that in a lighter blue, 4.4 FOSS. Tregs are in green, and we have mitosis in red that has high abundance, as well as a killer RB and MT over here, and then Ripple 32. And then the Pi-Lu evenness showing in each tissue, blood, normal, and tumor, per cluster, the immune repertoire ID. So I was looking at this and I found those 3.1 MT in blood, normal, I found 4.2, RPL 32, and tumor 4.8B Treg to be the most significant for the immunomine Pi-Lu evenness. And this Jacquard similarity index, or Tanimoto